Welcome back to Chapter 11, Part 2. We're going to cover DNA replication in eukaryotes and RNA transcription in this video. Previously, we discussed DNA replication in bacteria. Now let's compare that to eukaryotic replication, which is more complex. Eukaryotic chromosomes are linear and much larger, so replication has to start at multiple origins along each chromosome, not just one like in bacteria. Replication is also slower in eukaryotes, only about 100 nucleotides per second. This is about 10 times slower than what we see in prokaryotes. In eukaryotes, there are two key polymerases that handle this job. DNA polymerase epsilon primarily synthesizes the leading strand, and DNA polymerase delta primarily synthesizes that lagging strand. Notice that the book states the opposite, but this is based on most current research, and if things change, I will come back and edit this video and send out a notice. Finally, we have RNase H. This is going to be removing the RNA primers, in contrast to bacteria where we had DNA polymerase 1. And here we also have a ligase in eukaryotes to seal gaps. This is a nice figure that shows multiple replication bubbles forming across a linear chromosome, and each origin opens up in two directions, creating several replication forks that eventually meet and merge, and this coordinated system helps eukaryotic cells complete replication efficiently, even with billions of base pairs. Now let's look at what happens at the ends of linear chromosomes. These are called telomeres. And telomeres are repetitive, non-coding DNA sequences. That means they do not encode proteins, and they're usually repeats of TTA-GGG, repeated hundreds or even thousands of times at these ends of our chromosomes. These telomeres protect essential genes from being lost as the DNA shortens during replication, and the enzyme telomerase maintains telomere length by adding new repeats using an RNA template that it carries inside itself, and I'll show you that in a bit. Telomerase is active in germ cells, stem cells, and many cancer cells, but largely inactive in most body cells or somatic cells. When telomerase is not active, telomeres will gradually shorten, and that contributes to cellular aging. Elizabeth Blackburn won the Nobel Prize for her work on telomeres in 2009, and later on in around 2017, I believe, she published a book that emphasizes that lifestyle factors like stress levels, your diet, your exercise, sleep quality, and even your social environment can significantly influence telomere length. So this book is found pretty much at any public library. I believe we also have it at our college library, and it's written for non-scientists, so anybody can read more about telomeres if they're interested. So in DNA replication in eukaryotes with linear chromosomes, some of the ends of our DNA, our chromosomes, are lost with each round of DNA replication. Why is this? So during DNA replication, remember that DNA polymerase can only add nucleotides to an existing 3' end, right? That 3' hydroxyl group. It can't start a new strand on its own. So just a quick refresher, during DNA synthesis, replication requires a short RNA primer made by primase. And on the leading strand, this is not an issue, right? Synthesis is continuous all the way to the end. But on the lagging strand, remember DNA is made in short Okazaki fragments. I see that here and here, each starting with its own RNA primer. When that very last primer near the end of the 3' end of the template is removed, so here it's removed, it's also removed here, there is no upstream 3' hydroxyl group for DNA polymerase to use to fill in that gap. And as a result, the very end of the chromosome cannot be copied. So here we can see telomerase in action. It extends the 3' end of DNA using its built-in guide, its built-in sequence as a template, so we are going to protect coding DNA from loss. And remember, this is not present in all cells, so most body or somatic cells do not have active telomerase, That means with each round of replication, DNA does shorten, and eventually we do lose some coding DNA, some DNA that encodes proteins, which can lead to cell aging. Let's look at some key differences between bacterial and eukaryotic replication so far. We saw that in bacteria, they typically have a single circular chromosome, whereas in eukaryotes, we have multiple linear chromosomes that are much larger. The number of origins of replications in bacteria is 1. In eukaryotes, we have many. The rate of replication is slower in eukaryotes, and telomerase is only present in eukaryotes. Remember that DNA polymerase 1 was the one that removed the RNA primer in bacteria, whereas RNase H is the one that removes the primer in eukaryotes. And finally, the main polymerase responsible for elongating or making the new DNA daughter strands is DNA polymerase 3 in prokaryotes, and it's DNA polymerase delta and epsilon in eukaryotes. All right, our checkpoint asks, how does the origin of replication differ between eukaryotes and prokaryotes? So we just said prokaryotes have a single origin of replication in their circular chromosome, whereas eukaryotes have multiple origins of replication on each linear chromosome to allow faster genome duplication. For the second one, what enzymes are responsible for eukaryotic DNA replication? Remember, we have polymerase epsilon, which primarily synthesizes the leading strand, and polymerase delta, which primarily synthesizes the lagging strand. What's found at the ends of eukaryotic chromosomes? So we saw that we have telomeres. These are repetitive, non-coding DNA sequences found at those chromosome ends, and they protect coding genes from loss during replication. So the enzyme telomerase maintains telomere length in germ cells and stem cells and many cancer cells. Now let's look at a special form of replication used by plasmids, some phages, remember these are viruses that infect bacteria, and some small circular viral DNAs. So this is called rolling circle replication. It starts with an enzyme makes a nick in one strand of the circular DNA molecule. DNA polymerase number 3 then extends the 3' hydroxyl end of the nick strand, displacing the old strand as it synthesizes a new one. The displaced strand is then going to recircularize and serve as a template for a complementary strand, and each cycle is going to produce a new double-stranded copy of the original circular DNA. So here's the diagram for rolling circle replication. We see that nick here forming in one strand of the circular DNA molecule, then DNA polymerase 3 will extend the hydroxyl group of that nick strand, and that displaces the old strand. The displaced strand is going to then recircularize and be a template for a complementary strand, so each cycle is going to produce one new double-stranded copy of the original circular DNA. So our next checkpoint asks if there's a lagging strand in rolling circle replication, and the answer is no. There is no lagging strand in the initial rolling circle synthesis. The process is unicellular and it's continuous around that circular DNA template. One strand will be displaced, but it's later copied separately, but not in short Okazaki fragments, so no lagging strands. All right, let's recap 11.2. Remember, some of this information is from our first video, part 1. Explain the meaning of semi-conservative DNA replication. So semi-conservative replication means that each new DNA molecule contains one original parent strand and one newly synthesized strand, because when the double helix separates, each strand serves as a template for building its complementary strand. And this really ensures accurate inheritance of genetic information with minimal errors. All right, for the second one, why DNA replication is bidirectional, includes both a leading and lagging strand. So in most organisms, replication starts at a specific origin of replication and proceeds in both directions along the DNA, forming those two replication forks. Because DNA strands are anti-parallel, remember they're facing 5 to 3 and 3 to 5, and polymerase can only add nucleotides in the 5 to 3 direction, synthesis either occurs continuously toward the fork on the leading strand or discontinuously away from the fork on the lagging strand. For the third one, why Okazaki fragments are formed. So again, DNA polymerase can only synthesize or extend DNA in the 5' to 3' direction. So on the lagging strand, this direction runs away from the fork towards the center of that bubble. So synthesis must be started or restarted repeatedly as the fork opens up more and more. And each short segment of DNA is produced and called an Okazaki fragment, and these are later joined by DNA ligase to form a continuous strand. All right, so for the fourth and fifth learning objectives, I'm going to skip these for now, and I'm going to use a table to show you how you can practice these in a second. Let's go to the last one, the process of rolling circle replication. We said rolling circle replication happens in plasmids, bacteriophages, and circular viral genomes, and it happens when, or it begins when, an enzyme nicks one of the strands at a specific site. Then DNA polymerase 3 will extend that 3' end using the intact strand as a template, and it displaces the other strand as it goes. That displaced strand is going to eventually recircularize and serve as a template for complementary strand synthesis, and that's going to also produce a new double-stranded DNA copy. And this process is continuous and unidirectional. There's no lagging strand during the initial synthesis. So going back to that fourth learning objective, which said to describe the process of DNA replication and the function of the enzymes involved, we're really focusing on bacteria in that learning objective. Try to go back to that table. So I put one here, and I blanked out the functions of these enzymes and proteins. See if you can verbally or even practice writing them down to review this objective. And also try to do the same thing for learning objective 5. Identify the differences between DNA replication in bacteria and eukaryotes. I did the same thing, blanking out these boxes. See if you can go through these different properties and answer or fill in the blanks for each of these items. Now let's transition to section 11.3, which looks at RNA transcription. So here we're going to look at how the genetic information DNA is transcribed into RNA, and that's going to later direct protein synthesis through translation. Transcription converts DNA information into a single-stranded RNA transcript. RNA bases include A, C, G, and U, with uracil replacing thymine. And this process happens inside a transcription bubble, where DNA is temporarily unwound so the template strand can be copied. All right, so here's my original DNA molecule. And remember that only one strand is going to serve as the template strand because RNA will be single-stranded. The template strand is sometimes also known as the antisense strand. And the other strand, the non-template DNA strand, is often known as the sense strand. So it turns out the sense strand of DNA is going to be identical in sequence to the new RNA that is formed, except that in RNA we're going to have U's, uracils, whereas in the sense strand we're going to have T's, thymines. So I'm going to look more closely at that to show you how that occurs. So let's just look from here to around over here. So just that segment of my sense strand comparing to my RNA strand that I'm just forming right now, I can see that instead of T's, I have U's, and the rest of this is identical. CAC, GCA, so CAC, GCA, and then CTCA. I have CUCA instead, because remember there's no T's in my RNA. So they're going to be identical in sequence again, except for those T's and U's, although in our book, in this picture, if you look very carefully, there are some typos. Don't worry about those typos. Know that the sense and the RNA strand should be identical except for that U versus T. And RNA is going to be synthesized also from the 5' to 3' direction. You can only add nucleotides to the 3' end by RNA polymerase, which adds complementary ribonucleotides one at a time. In bacteria, only one type of RNA polymerase handles all transcription, and unlike DNA polymerase, it does not require a primer. There is a sigma factor that helps RNA polymerase recognize a region called the promoter, a specific DNA sequence that signals where transcription should start. And once RNA polymerase binds the sigma factor will be released and elongation begins. So relative to RNA, DNA is very big. So how do we know which part of the DNA we want to transcribe and produce RNA? It turns out before the RNA molecule is transcribed, we have to look for a region on the DNA sequence called the promoter. So the promoter tells you where transcription is going to begin. And transcription starts at the +1 site. This is where you put down your first RNA nucleotide. So if I were to look down at this picture, our +1 site looks like right here. That is +1. That's where I put down my first RNA nucleotide. Anything to the left of that, before that, is known as upstream, and anything after that is known as downstream. So over here would be upstream, to the right would be downstream. Promoters are always going to be upstream of our +1 site, and we count backwards. So right before or upstream of +1, we have -1, -2, -3, and these are individual nucleotides, each number. There's no 0 here, so we go straight from +1 to -1. If we had more detail in this picture, we would see that at the -10 site, there would be a region with a conserved sequence, TAAT, often called the Tata box, and there would be another conserved promoter sequence at -35. So if I were to keep going all the way to -35 as well. These conserved promoter sequences, where conserved means these are DNA sequences that have stayed very similar or identical across different species, different genes, or even over evolutionary time, they are recognized by a part of the polymerase known as the sigma factor. Right? So the sigma factor at first is complexed or bound to RNA polymerase, and the sigma factor binds to the -10 and -35 sites to ensure correct positioning of RNA polymerase. And that picture from our book didn't really show the promoter region, so I wanted to give you another reference, but don't worry about the details. That sigma factor first binds to the -35 region of the promoter, and that step allows RNA polymerase to anchor and position itself correctly on the DNA. After anchoring at -35, the sigma factor interacts with that -10 at Tata box region, and that Tata box region is AT rich, right? These only have 2 hydrogen bonds compared to G's and C's, making this area of the DNA easier to unwind. So RNA polymerase, guided by sigma, will eventually unwind or melt that region, breaking those hydrogen bonds, forming the transcription bubble. So now we're ready to begin elongation of RNA. After initiation, the sigma factor will dissociate, and the core RNA polymerase enzyme will continue elongation, synthesizing RNA in that 5-3 direction. And this will happen at around 40 nucleotides per second. That's the rate. As RNA polymerase moves forward, DNA is going to unwind ahead and reform those hydrogen bonds behind. So termination is when transcription ends, and transcription ends when RNA polymerase encounters termination signals in the DNA. There are 2 main mechanisms, either intrinsic, which is also known as row-independent termination. This involves an RNA hairpin loop, followed by a stretch of U's that causes RNA polymerase to pause and release, or row-dependent termination, which requires a row protein. This binds to the RNA, moves along it, and helps detach the transcript, that RNA transcript, from the DNA. In both cases, the result is a newly formed RNA molecule that separates from the DNA template. So those are shown here. We have intrinsic or row-independent termination. What happens is there is going to be a bunch of G's and C's in our RNA transcript, forming this hairpin loop, followed by a bunch of U's because the template will have a bunch of A's. This big bulky structure, followed by weaker, remember only 2 hydrogen bonds between A's and U's, will cause this whole molecule, the RNA molecule, to fall off and separate from the template strand. In row-dependent termination, shown here, there's a donut-shaped protein, that row protein, that threads itself through the RNA, and that will eventually separate RNA from the template strand. So where does sigma factor of RNA polymerase bind to DNA to start transcription? The sigma factor binds to the promoter sequences on DNA, which are just going to be upstream of the gene of interest, that gene that will be transcribed. So specifically, sigma factor recognizes two conserved regions on DNA, the -10 region, that Tata box, and the -35 region. So binding at these promoter sites will correctly position RNA polymerase to begin transcription at that +1 start site. For the second one, what occurs to initiate polymerization activity of RNA polymerase? So kind of similar to the first one. Once RNA polymerase is bound to the promoter, DNA unwinds, forming that transcription factor. The sigma factor will be released, and that activates the core enzyme, the core RNA polymerase enzyme. RNA polymerase can start adding ribonucleotides to that growing RNA strand in that 5' to 3' direction. So remember, no primer is needed. RNA polymerase can start from the beginning. And finally, where does the signal to end transcription come from? So the DNA template will contain specific termination sequences, and these sequences cause RNA polymerase to stall and detach from the DNA. That will release the RNA transcript, and that marks the end of transcription. Now let's switch gears and look at transcription in eukaryotes. It's more complex and will occur inside the nucleus. So later we'll also see translation, the process of going from mRNA to protein, will happen in the cytoplasm. But transcription in eukaryotes uses 3 RNA polymerases. RNA polymerase 1 transcribes ribosomal RNA genes, RNA polymerase 2 transcribes protein coding genes, and RNA polymerase 3 transcribes transfer RNA and small RNAs. Eukaryotic messenger RNAs are monocistronic, meaning one messenger RNA codes for one protein, unlike bacterial mRNAs, which can be mono or polycistronic, and that means one messenger RNA encodes for multiple proteins. So just using that textbook picture as an example, if this were in a eukaryote, this messenger RNA, if it were a messenger RNA, would just encode one protein. That would be monocistronic. But if this was in a bacterium, it could either be monocistronic or polycistronic, where maybe one segment encodes one protein and a different segment encodes a different protein. So that would be polycistronic. Unlike prokaryotes like bacteria, where as soon as messenger RNA is created, you can translate that into protein, in eukaryotes, we have to undergo several steps of RNA processing. So the first thing that is created in eukaryotes is actually pre-messenger RNA. That is the primary transcript, or the RNA molecule that was created from the DNA template. So 3 things that have to happen include 1, adding a 5' G cap. So if this were my messenger RNA, messenger RNA, I would have to add a G cap, a 5' G cap on the 5' end, a poly A tail, so a bunch of A's on the 3' end, and these help prevent degradation of my messenger RNA. So what happens inside our cells is that we want to make sure we don't have viral RNA in there, right? A lot of viruses are RNA viruses. So our enzymes, if we just see free-floating RNA out there, might just chop it up in case it's viral. To protect our messenger RNA so that there is time for it to be stable and translated accurately in the cytoplasm, we have to add the 5' cap and the poly A tail. So again, that prevents degradation. The 5' G cap also allows for translation later. Ribosomes, we're going to see in the next video, bind to the G cap and begin translation. So again, these modifications will stabilize our mRNA so that we can translate it later in the cytoplasm. And our third step of RNA processing is here, RNA splicing. So it turns out that if this were my pre-mRNA molecule, 5' to 3', and I had that G cap and that poly A tail here in those first 2 steps, there are regions in my messenger RNA that are coding for proteins, but there are also regions that are non-coding. So I'll put the non-coding in blue. So introns are intervening sequences that are non-coding. They don't encode for proteins, whereas exons do. What happens in RNA splicing is that these introns will be removed, and that's completed by these enzymes called spliceosomes. We'll see that they contain small nuclear ribonucleoproteins as well. Exons will be joined together to form a continuous coding sequence, and I'll also show you how introns can be spliced in different ways, called alternative splicing, to generate different messenger RNAs from a single gene. Finally, when all three steps are done, the GCAP, the poly A tail, and the splicing, the mature messenger RNA is ready to leave the nucleus and enter the cytoplasm for translation. These next three pictures come from a different text, the Biology 2E Openstax book, but it nicely summarizes many of these components. So here we have the three steps of RNA processing, our 5' GCAP, our poly A tail, and they show the splicing out of those introns and joining together of those exons. Remember, exons are expressed. The second figure shows splicing, so I'm cutting out the intron and joining the exons together, and it shows you a picture of a spliceosome. Spliceosomes include snRNPs, which I mentioned earlier, small nuclear ribonucleoproteins, and they are kind of these egg-like structures, and they themselves contain small RNA molecules. So spliceosomes are going to recognize splicing sites, and they cut out introns and join exons together to form mature mRNA. And here's a look at that alternative splicing, where you can take one gene and make multiple versions of messenger RNAs. So if this is my pre-mRNA, here they splice out the introns on the left side, but they also have removed this one. So you can see when they're cutting this out, they also removed that whole section for this messenger RNA. On the other side, let me erase this really quick. On the other side, if I'll use a different color, they are cutting out the introns here as well, but I don't see exon number 2, so they also remove this exon, and they're only splicing together genes 1, 3, and 4. And since eukaryotes have a nucleus, remember that all of this is currently still happening in the nucleus. So transcription took DNA and formed our pre-messenger RNA. The RNA processing is also happening in the nucleus. Once those three steps are done, the GCAP, the poly A tail, and the splicing, now we have our mature messenger RNA that can exit the nucleus and enter the cytoplasm for translation to occur. And our book has a great table comparing and contrasting transcription in bacteria versus eukaryotes in terms of the number of polypeptides encoded per messenger RNA, the polymerases involved. In blue, those rows I added in. So we saw that only eukaryotes need to process their messenger RNA. We added that 5' GCAP, the poly A tail, and splicing out of the introns. This processing actually lengthens the stability of our transcript. So messenger RNA lasts for hours in eukaryotes versus seconds in prokaryotes like bacteria because they don't have these protections. So enzymes will chop up the RNA quickly. Location, remember this happens in the nucleus for eukaryotes, but everything happens in the cytoplasm of bacteria. Our checkpoint asks, in eukaryotic cells, how is the RNA transcript modified? We had our 5' GCAP, our poly A tail, and we spliced out the introns through those spliceosomes, and we formed a mature messenger RNA that will be transported to the cytoplasm for translation. For the second one, exons or introns, which one codes protein sequences? It's the exons. So exons contain the coding information. Introns are non-coding regions. All right, let's recap 11.3. So how is RNA synthesized? Remember, during transcription, the DNA double helix unwinds in a region called the transcription bubble, and only one strand, the DNA template strand or antisense strand, is used to make the RNA. RNA polymerase reads the template in the 3' to 5' direction and builds the RNA in the 5' to 3' direction, adding complementary ribonucleotides like A, U, C, and G to form that RNA transcript. Remember, U or uracil pairs with adenine instead of thymine. So our finished product will be a single-stranded RNA molecule that is complementary to the DNA template. For the second one, I'm going to use a table like I did before to help us distinguish prokaryotes and eukaryotes. Please use these empty boxes to practice that second learning objective. distinguish between transcription in prokaryotes and eukaryotes, and then check your answers by going back to that previous table. That takes us to the end of Part 2. In Part 3, we're going to cover Section 11.4 on translation.